Male and female reproductive system. So let's take a look at the female reproductive system first. This is a section through the ovary. The ovary has two major portions. You have the outer portion and the muscular and highly vascularized middle portion, the medulla, so cortex and medulla. Here's a higher magnification view. You can see the thick connective tissue layer called the tunica albuginea. Just outside that layer you can see an epithelium composed of simple cuboidal cells. This layer is called the germinal epithelium, which is actually a misnomer because these are not germinal cells. It was once thought they were, so it still retains the name. Inside the cortex of the ovary you can see numerous cell types. Most of these are follicular cells. Most of these cells are follicular cells. You can see the presence of large oocytes, which are these larger cells with large nuclei and condensed chromatin inside those. These are the oocytes. You can see that these particular oocytes are surrounded by squamous follicular cells around them, and so these are referred to as primordial follicles at this stage, and this represents the first stage of follicular development. On this slide you can see the oocyte is surrounded by the follicular cells called granular cells, this stage, which are more cuboidal in shape. And at this stage, this follicle, which includes both the oocyte and the follicular cells, is a primary follicle. You can also refer to it as a unilaminar follicle because it has a single layer of follicular cells. Here is a follicle further on in development, and you can see the oocyte here. At this point you can see the follicular cells, which are cuboidal in shape here. There is more than one layer. There are several layers. So this is what we would call a growing follicle or a multilaminar follicle. You can also see at this stage the presence of a glycoprotein barrier called the zona pellucidum that surrounds the oocyte, and so is between the oocyte and the deep layer of the follicular cells. You can also see at this stage there are other follicular cells that are beginning to aggregate and encircle around the follicle in the outer layers. These remain flat, and these cells are called thecal cells. So you have the granulosa follicular cells and the thecal follicular cells. Here's a later stage of follicular development. At this stage you can see there are these pockets that are forming between the granular follicular cells. This is called an antral cavity. These cavities are filled with antral fluid, so this stage of a follicle is said to be an antral follicle. This is an early to mid stage antral follicle. As the follicle begins to develop further, more and more of the volume is occupied by this fluid-filled antral cavity. You can see here, because of the accumulation of fluid in the antral cavity, that the oocyte now is only connected to part of the wall of the follicle by granulosa cells. So you can see these cellular bridges that are connecting the oocyte over here to this wall. The region where the cells are attaching the oocyte to the wall of the follicle is known as the cumulus oovarus, and the innermost layer of the cumulus oovarus, immediately adjacent to the oocyte, is called the corona radiata. You can see here that in this stage of the antral follicle that the thecal cells are becoming well defined, and you can actually begin to see two layers, an inner and outer layer of thecal follicular cells. This is a late stage antral follicle which occurs fairly soon before ovulation. You can see that most of the volume of the follicle is now occupied by this very large antral cavity. The oocyte is displaced off to the side of the follicle and is still attached to the wall of the follicle via the cumulus oophorus. You can see the corona radiata that is especially well defined in this particular specimen. This is the last stage of the follicle that we have on a slide to show you. The latest slide that's possible is a Graafian follicle. The Graafian follicular stage is relatively short because it immediately precedes ovulation, and that's when you have an antral follicle that's so large it is taking up most of the cortex and is actually causing the outer portion of the follicle to bulge out. So we don't actually have a slide of an antral follicle or of a Graafian follicle. This is as close as it gets. So this is a late stage antral follicle. There is a model in the lab where you can see the Graafian stage. So the Graafian follicle stage is a stage that immediately follows ovulation. During ovulation, the outer portion of the ovary ruptures, so the capsule breaks open. The oocyte, along with some of the cumulus oophorus, is expelled into the peritoneal cavity of the female for a very short time before it's swept up into the oviduct or fallopian tube. What's left behind is essentially the husk of the follicle, which quickly begins to differentiate and form a temporary endocrine organ, which is known as the corpus luteum. Corpus luteum means yellow body. It gets its name because it appears yellowish in fresh specimens. Oftentimes there's rupture of blood vessels, which cause a formation of a blood clot inside the lumen of the corpus luteum, which you can see here. And that structure is called a corpus hemorrhagicum. Here's a higher magnification view of the wall of the corpus luteum, and as the cells differentiate, there are two major populations of cells that form the two major parts of the cell wall. Both of these secrete different female hormones, such as estrogen and progesterone. Most of the width of the cell, the inner cells, are called granulosa lutein cells, or simply lutein cells. The more basophilic cells that you can see towards the outside represent thecal cells that have differentiated and are now called thecal lutein cells. So granulosa lutein cells, or simply lutein cells, are the same thing. And then you have the thecal lutein cells. Thecal lutein cells, again, are found towards the outside, and the wall of the corpus luteum typically bends, and you can see those in the folds of the outer bends. Here is a H&E stain section that shows the thecal lutein cells particularly well because they are quite a bit more basophilic than the lutein cells. So make sure you can identify both those types of cells in your sections of the corpus luteum. If there is no pregnancy or implantation of fertilized egg or developing embryo onto the uterus, then there is no hormonal feedback to sustain the corpus luteum, and it degenerates. When it degenerates, it gets infiltrated by fibroblasts, which forms this scar tissue structure, which is known as the corpus albicans, which means white body because of its white fibrous appearance. This is a section through the distal portion of the fallopian tube or oviduct, the portion that is closest to the ovary. This part of the oviduct, here you can see the lumen, which is highly folded, and this part of the oviduct is called the infundibulum because of its flared opening. Infundibulum means funnel shape, and you can see these finger-like projections here, which are called fimbriae, and these fimbriae possess ciliated and non-ciliated epithelium. The movement of the cilia beating helps to sweep the oocyte into the oviduct, where fertilization typically occurs. Here's a section probably from the middle section of the oviduct, which doesn't have quite a flared opening, but it is wider than the section closest to the uterus. So this section would be called the ampulla. The section closest to the uterus, the proximal segment, is referred to as the isthmus and is very narrow. So here's a section of the uterus. The uterus has three major layers to it. You have the innermost layer, which is called the endometrium. The thick middle layer is called the myometrium, and is composed of smooth muscle. And then finally, the outer third layer is called the perimetrium, and it is composed of connective tissue. You cannot see the perimetrium on the slide. It would be out here. So here's a higher magnification view of the endometrium. The endometrium itself has two major layers. You have the thick stratum functionalis, or functional layer, and then you have the lower stratum basalis. The stratum functionalis is highly vascularized, and it contains many glandular ducts, which are relatively tubular in shape. You can see here. This is the early stage of the menstrual cycle, which is known as the proliferative stage, where the cells in the stratum basalis are proliferating to regenerate the stratum functionalis after it's lost in menses. So this is what the endometrium looks like during the earlier proliferative phase. After the proliferative phase, cell growth begins to slow down. The cells are less mitotically active, and the stratum functionalis begins gearing itself for secretion of nutrient-rich secretions to potentially support an implanted conceptus. And so this would be the early secretory phase of the endometrium, which is characterized by the glandular ducts becoming less straight tubes and more spiral in shape. So where you can start to see these spiraling glandular ducts, as well as the capillaries in the wall of the endometrium also begin spiraling as well. This is a section of the endometrium from later secretory phase, and you can see that there is an even greater bending to the glandular ducts, such that they're somewhat S-shaped or sigmoidal shaped in many cases. So this is the late secretory phase of the endometrium, which immediately precedes menstruation if there is no attachment of an embryo. So if there's no attachment of an embryo, then as a result of a lack of hormonal feedback, the vasculature in the endometrium starts to kink and spasm. Eventually, they cut off the blood supply to the upper layers, or superficial more layers here, so these layers become ischemic, and this tissue ends up dying and becomes sloughed off in the menses. So that's what this slide is showing you here. This is endometrium during the menstrual phase. It has a very ragged appearance superficially. Most of the functional layer is sloughed off in the menses, and mostly what remains is the stratum basalis, which begins proliferating during the proliferative phase. And then finally here we have a section of the vagina of the female, which is characterized by having a stratified squamous non-keratinized epithelium. So that's it for female reproduction. Let's talk about male reproduction now. This is a section through the testes. The testes has a lobulated appearance, meaning that it is divided into discrete functional lobules, which are divided by septum filled with connective tissue. So here you can see these connective tissue septa. On the outside of the testes, there's a thick layer of connective tissue, which is called the tunica albuginea, just like the outside layer of the female ovary as well. Here's a higher magnification view where you can see the tunica albuginea and the inside one of the lobules of the testes, which is filled with many tube-like structures called seminiferous tubules. Seminiferous tubules are the sites of spermatogenesis or sperm production. Here's a higher magnification view of several seminiferous tubules here. You can see at a low magnification view, they really have, or a medium magnification view, they really have a polka dotted appearance. All of these dots are actually the dense basophilic nuclei of various states of spermatocytes and various states of development. Here's a higher magnification view of several seminiferous tubules. There are lots of spermatocytes in multiple stages of development, but there's three major classes of cells you need to be able to identify in the seminiferous tubules for this lab. The first cell type are called spermatogonia, which look like many other spermatocytes in that they have a very regular, round, spherical or round appearance with very dense basophilic nuclei. But these cells are located at the basal lamina and the basal surface of the seminiferous tubules. These cells are called spermatogonia. Spermatogonia are precursor cells that give rise to later stages of spermatocytes. You can see spermatocytes in various stages of development. Here are some early stage spermatocytes. Further up, you can see some later stage of spermatocytes where they are beginning to look more like mature spermatozoa. At this stage we would call these spermatids. There are early and late stage spermatids. Just identify any of these cells for the purpose of this lab. You just need to identify them as spermatocytes. So spermatogonia are found at the basal surface of the seminiferous tubules attached to the basal lamina. Spermatocytes are found at the more apical layers. The third type of cell that you need to identify are called Sertoli cells. Sertoli cells are very large cells that span the entire width of the wall of the seminiferous tubules. So they attach to the basal lamina and they actually go all the way up to the apical surface, although it's usually hard to see where their cytoplasm spans the entire width. The best way to identify Sertoli cells is by looking at their nuclei. Unlike spermatocytes and spermatogonia, their nuclei tend to be more ovoid or elongated instead of spherical in shape, and they have very prominent dense nucleoli, which are those little dot-like appearing structures in the nucleus, as opposed to spermatogonia and spermatocytes, which do not have visible nucleoli. So to be able to see these cells and distinguish them from other cells, you really need to view testes at a magnification of 400x. Here's a higher magnification view. Again, you can see here's a Sertoli cell with its nucleus. Here's two more Sertoli cells. This is all Sertoli cytoplasm. It's tough to see where one cell ends and one begins. They actually go all the way up to the apical surface, although it's hard to see that because of the presence of all the other spermatocytes around them. These are all spermatocytes in various stages of development, and here you have spermatogonia. Here are two more spermatogonia. So make sure you can identify all those three classes of cells within the seminiferous tubules. There are also important cells in the interstitial spaces between the seminiferous tubules. Some of these cells are endocrine cells, and these are called interstitial cells of Leydig. These cells are responsible for secreting most of the androgens in males. So these cells produce testosterone. Much of the testosterone leaves the testes by diffusing into nearby capillaries. Some of it diffuses locally into the seminiferous tubules, where the testosterone acts to boost sperm production. So testosterone is absolutely essential to maintaining optimal sperm production. And this is further boosted by secretion of a molecule by the Sertoli cells, which is known as ABP, or androgen binding protein. The androgen binding protein helps to concentrate the testosterone that's secreted by the interstitial cells, further increasing its activity in the testes. This is a section of the next organ in male reproduction. This is the epididymis. So the epididymis is a separate organ from the testes. So sperm are produced in the seminiferous tubules, and they don't fully mature in there. Rather, they pass through the rete testes and then enter into this structure known as the epididymis, where they continue to mature until they become motile and ready to fertilize an egg. So epididymis at low magnification has a fairly similar appearance to the testes. It is lobulated, so you have these connective tissue septa dividing lobules of the epididymis, and you have this outer connective tissue layer here as well. And you also have lots of tube-like structures in cross-section. A difference is that the epididymis is actually one very long, highly coiled tube. So all of these tubular sections are all different sections to the same very long coiled tube, whereas seminiferous tubules are all independent tubules. Here's a higher magnification view of the epididymis, and you can see a major difference between the walls of the epididymis and the testes. The testes have that polka dotted appearance due to the presence of many layers of spermatocytes. The wall of the epididymis is composed of pseudostratified epithelium, and on the apical surface you can see specializations which look like cilia because they're these long hair-like processes, but these are actually called stereocilia, and you may recall from earlier on in the trimester that stereocilia look like cilia, but they're actually long branched specialized microvilli. So these contain an actin core rather than a core composed of axonemes. And going back real quick, you can often see some sperm either loosely distributed throughout the lumens of the sections of epididymis. Sometimes you can see it clumped up as a precipitate. Sperm continues to mature in the epididymis, so the more distal portions of the epididymis have sperm that are more mature and ready to fertilize an egg. They remain there in storage until ejaculation, after which they pass through the lumen of this structure. This is known as the ductus deferens, also known as vas deferens. So the vas deferens is a long tubule that connects the epididymis with the ejaculatory duct of the penis. So sperm has to pass through the narrow lumen of this structure. It's propelled through here by rhythmic contractions of the very muscular wall, which is composed of many layers of smooth muscle that contract rhythmically during ejaculation. The common procedure, which is known as a vasectomy, involves cutting this structure, which prevents sperm from leaving the testes. So in a vasectomy, the testes are left intact, sperm are still produced, but they cannot enter the ejaculatory duct because this structure is cut. There are numerous accessory glands that contribute to the volume of semen. Semen's a complex mixture you'll learn about in lecture. We're only going to look at one of the accessory glands in lab, which is the prostate gland. Most of the volume comes from the seminal vesicles, but a significant volume of the semen comes from prostatic secretions. So the prostate has a fibromuscular stroma, and here you can see many ducts of glands. This is a prostate from an older male, so you can see the presence of these spherical shaped prostatic concretions, which tend to accumulate as a male ages. They are not typically associated with any sort of pathology. This is what the prostate looks like from a younger male, and note there are no visible prostatic concretions in the lumens of these ducts here. And then finally, we're going to look at a cross-section of the penis. We saw this for the previous lab where we focused on the male urethra. So this is the male urethra. Recall that the male urethra is surrounded by highly vascularized tissue known as the corpus spongiosum. You can see two other highly vascularized tissues on the dorsal surface of the penis. These are called collectively the corpora cavernosa. These are also highly vascularized, and in the middle of these structures they contain two major arteries called the deep arteries that cause the penis to fill with blood and engorge as blood perfuses through the vascularized tissue surrounding the deep arteries. Those arteries are cut off in this particular section, but you may be able to see them on your slides. So that's it for lab this week, and that's all.